today we are going to learn about the gene delivery systems, one of the most exciting topics in the modern biotechnology and the pharmaceutical science. So before we start, I want to ask you a question. Suppose your mobile phone suddenly stops working, so because its software is damaged, so what will you do? So most of you would update the software or reinstall it. So our body also has a kind of software. So that software is called the genes. Okay. So genes are made up of DNA and are present inside the nucleus of every cell. So they contains the instructions that tell our body how to grow, how to produce the proteins, how to repair the tissues, and how every organ should function. So sometimes due to inheritance or mutation, so these genes becomes defective. So when this happens in disease state, like in hemophilia, thalassemia, cystic fibrosis, and even some cancers can occur. So early medicines only reduce the symptoms of the diseases here. So but now scientists are trying to correct the defective gene itself. So to do this, they need a way to carry a healthy gene into the patient cell. Again, I am telling, to do this, they need a way to carry a healthy gene into the patient cells. So this process is called the gene delivery. So today's class will explain what gene delivery is, why it is needed, so different delivery methods, viral and non-viral systems, applications, future developments. So by the end of this lecture, you will understand how scientists can repair the body's genetic software. So what is gene delivery? Now look at this slide. On the left side, you can see a DNA molecule. On the right side, there is a human cell. So gene delivery means transporting the DNA, RNA, or gene editing molecules into the target cell so that the cell can produce the correct protein. Okay, so let us understand this using a simple example. So imagine your parents wanted to send you a parcel because you are staying in a hostel. So for successful delivery, 4 things are needed here. 1 is the parcel, 2 a courier service, 3 the correct address, and 4th is the person receiving the parcel. So the same concept applies to the gene delivery also here. So the gene is the parcel. Okay, the gene itself is the DNA, and the vector, the virus or the nanoparticles, is the courier, and the target cell is the address, and the cell nucleus is the receiver here. So if the parcel reaches the wrong address, it is useless. Similarly, if the therapeutic gene enters the wrong cell, the treatment will not work and may even cause side effects also. So that is why the scientists focus on the targeted gene delivery here. Example, so imagine a patient with a defective gene responsible for producing the insulin. So normally the patient takes insulin injections every day. So instead, the scientists are trying to deliver a healthy insulin producing gene into the pancreatic cells so that the patient own body starts producing the insulin naturally. So that is the basic idea behind gene therapy. Okay, so need for the gene delivery. So now let us understand why the gene delivery is necessary. So the human body contains approximately 20,000 genes. So every gene performs a specific function. So if one important gene becomes defective, the normal functioning of the body is get disturbed. So this can lead to diseases such as thalassemia, hemophilia, cystic fibrosis, sickle cell anemia, some inherited cancers. So the traditional medicines usually control the symptoms here. For example, the painkillers reduce the pain, insulin lower blood sugars, and the antibiotics kill bacteria, like that. So however, these medicines do not correct the defective genes. So real life analogy is, imagine there is a leak in the roof of the house. You place a bucket under the leak to collect the water here. So does this solve the problem permanently? No. So the real solution is repairing the damaged roof. So similarly, the medicines are equal to the bucket, that is temporary solution. The gene therapy is the repairing the roof solution, that is permanent solution. So the gene delivery allows the scientist to repair or replace the faulty gene itself here. So that is why the gene delivery is considered as one of the most promising technologies in the modern medicines. Okay. Components of the gene delivery. So now let us understand the 4 essential components of the gene delivery system. First one is the genetic material. So this is the therapeutic DNA or RNA. So it contains the correct genetic information needed to replace or repair the defective genes here. So think of it as a new instruction manual. Delivery vector, that is the second component. So DNA cannot enter the cell on its own because it is large and easily destroyed. So therefore it requires a carrier called the vector here. So the vector may be viruses, liposomes, nanoparticles or polymers. So whatever may be. So their function is to protect the DNA and transport it safely to the target cell. Okay. So think of the vector as a courier service carrying an important package. And third one is the target cell. So the vector delivers the gene only to the required cells. Example, liver cells, muscle cells, blood cells, brain cells. So different diseases require different target cells. And fourth one is the cell nucleus. So once the DNA enters the nucleus, it starts producing the required protein. So this protein helps restore the normal body function here. So the complete pathway is healthy gene, vector, target cell, nucleus, protein production and the disease improvement. Viral gene delivery systems. Now that we know that gene delivery is, so the next question is, how do we deliver the gene into the cell? So one of the most efficient method is the viral gene delivery. So when you hear the word virus, so what comes to your mind? So most of us think of diseases like COVID-19, influenza or dengue. So but the scientists have turned the viruses into a useful tool here. How? So a virus naturally enters the human cells to deliver its own genetic material. So the scientists remove the harmful gene from the virus and replace them with a healthy therapeutic gene here. So now the virus can no longer cause the disease. So instead it becomes a carrier or a vector that delivers the healthy gene into the patient's cell. So look at the image. So the virus first attaches to the cell surface, enters the cell and releases the therapeutic DNA. So the DNA reaches the nucleus where the cell starts producing the required protein here. So imagine a courier company delivering an important medicines to your home. So the medicine is equal to therapeutic gene here. So think like that. And the courier is equal to the virus and your house is equal to target cell. So the courier does not use the medicines. It simply delivers it safely. Similarly, the virus only transports the healthy gene here. So here I am asking one question. So can viruses naturally enter cells? The answer is yes. Exactly. So that is why scientists use them as a genetic carrier. Okay. So types of the viral vectors. So there are different viruses used in the gene therapy. So each has its own advantages also. So let us see about that. The first one is the adenovirus. So adenovirus delivers the gene very quickly. So it can infect many types of cells. So however, the gene expressions last only for a short time. So it is commonly used in the vaccine development and the cancer research also. And the second one is adeno-associated virus, that is AAV. So AAV is one of the safest viral vector here. So it causes a very little immune responses and produces a long-lasting gene expressions. Many approved gene therapy products use AAV. Okay, so AAV is equal to almost always very safe. That is simple memory trick here. And third one is the lentivirus. So lentivirus can insert the therapeutic gene into the cell's chromosome here. So this allows the long-term expression. So it is widely used in the stem cell therapy and some cancer treatments. And fourth one is retrovirus. So retrovirus also integrates into the DNA but mainly infects the dividing cells. So it is useful in certain blood disorders. And fifth one is the herpes simplex virus, HSV. HSV has a special ability to infect the nerve cells. It is used in the research for treating the neurological diseases such as Parkinson's disease. So again I will ask one question here. Which viral vector is considered the safest? The expected answer is the AAV. Okay. Mechanism of the viral gene delivery. So now let us understand how the viral gene delivery works. So look at the diagram. First step is attachment. So the virus recognizes the specific receptors on the surface of the target cell and attaches to it. So think of this as a person finding the correct house before delivering a parcel. And second one is entry. So after attachment, the virus enters the cell through the cell membrane. So it is like opening the front door of the house. And third one is releasing of the therapeutic DNA. So inside the cell, the virus releases the healthy DNA. So the virus has completed its job. Okay. And fourth step is gene expression. So the DNA enters the nucleus and the cell reads the new genetic information and starts producing the required protein here. So as a result, the defective gene is corrected and the normal cell function is restored here. So the real life analogy is, suppose a teacher gives you a new textbook. So you read the textbook and learn the correct information. Similarly, the cell reads the new DNA instruction and produces the correct protein here. So which organelle contains the DNA after delivery? That is nucleus. Advantages of the viral vectors. So why a viral vector widely used? So because they have several important advantages, like the first one is the high transfixion efficiency, that the virus naturally enters the cell very efficiently. So a large number of cells receives the therapeutic genes here. And second one is the long-term gene expressions. Some viral vectors such as Lentivirus, AAV continue producing the proteins for months or even years. So this reduces the need for repeated treatment. And third one is the target specificity. So certain viruses infect only the specific type of cells. So this improves the treatment and reduces the side effects here. And fourth one is the effective in the different cell types. So viruses can deliver the genes into both dividing and the non-dividing cells. So this makes them useful for treating many diseases here. Example, so think about two delivery services. One delivers your parcel safely every single time and the other loses half of the parcels. So which one would you choose? So obviously the reliable one. So viruses are like highly efficient delivery professionals here. Okay. Non-viral gene delivery systems. So far we have learned about the viral vectors. So although viral vectors are highly efficient, they also have some limitations such as immune reactions, limited gene size, and the high production cost. So because of these limitations, scientists develop non-viral gene delivery systems also. So point to the slide. So look at this image. So instead of using the viruses, scientists use the artificial carriers such as liposomes and the nanoparticles to transport the DNA safely into the cell. So these carriers protects the DNA from enzymes present in the body. So once they reach the target cell, they release the DNA inside the cell. Okay. So why are non-viral systems important. So they are safe, they produce a low immune responses, they are easy to manufacture, and they are less expensive, and then they carry a large DNA molecules. Okay, so the real life example is, imagine you want to transport a fragile glass items. So would you throw it into a truck without protection? No. So you wrap it in a bubble wrap. Similarly, so liposomes and the nanoparticles protects DNA until it reaches the target cell here. So if non-viral systems are safer, why do we still use the viral vector? So the expected answer is the viral vectors have higher efficiency. Chemical methods of gene delivery. So now let us discuss the most common chemical methods, that is liposomes. So look at the first image. So liposomes are the tiny spherical vesicles made of lipids here. So since the cell membrane is also made of lipid, so the liposomes easily fuse with the cell membrane and releases the DNA inside the cell. Okay, example, think about a soap bubble carrying in a small object inside it. So the bubble reaches the destination, bursts, and releases the object. So the liposomes work in the similar way here. The second one is the polymeric nanoparticles. So these are the biodegradable particles made from polymers. So they protect the DNA and releases it slowly after entering the target tissue here. Example, like a slow release medicine tablets. And dendrimers. So dendrimers have a tree-like structure with many branches. So these branches hold the DNA molecules firmly and improves the delivery efficiency here. Lipid nanoparticles, LNPs. So this is one of the most important modern delivery systems. So do you remember the COVID-19 mRNA vaccines? So they use the lipid nanoparticles only. So the mRNA was enclosed inside the lipid nanoparticles because naked mRNA is destroyed very quickly inside our body here. So how many of you receive a COVID vaccine? So most students will raise their hands. That means all of you have already experienced the non-viral gene delivery technology. Okay, so physical methods of the gene delivery. So far we discussed the viruses and the chemical carriers. So now we will study the physical methods where the physical forces helps DNA enter the cell. That is electroporation. So scientists apply a very little electrical pulse, that is very short electrical pulses. So that electrical pulse creates the temporary pores in the cell membrane. After that, the DNA enters through the pores. After a few seconds, the pores will close. Okay, so example, imagine opening a gate for a few seconds to allow someone to enter. So the gene gun. So this method is mainly used in plants. So tiny golden particles coated with a DNA are fired into the plant cells. So students usually smile here. So yes, it is literally called as a gene gun. So micro injections. So using a microscopic needles, DNA is injected directly into the single cell. So this method is highly accurate but time consuming one. And ultrasound mediated delivery. So ultrasound waves temporarily increases the cell membrane permeability here. So the DNA enters more easily here. So which physical method uses electricity? Tell me. That is electroporation. So which method uses the microscopic needles? That is microinjections. So like that we have to study. Viral versus non-viral gene delivery. So now let us compare both the systems here. Viral delivery. So what are the advantages of this system? Means here very high efficiency, long-term gene expression, and excellent targeting. So what are the disadvantages? Means having a high cost and the possible immune responses and a limited DNA size. Okay, and non-viral delivery. So what are the advantages of this system? Means these are safer, low toxicity, low immune response, easy manufacturing, and the larger DNA capacity. Disadvantages are lower efficiency and usually temporary gene expressions. Summary. So students, neither system is perfect. So if you need high efficiency, viral vectors are preferred. If safety is the most main concern, and non-viral systems are more chosen here. So researchers are continuously improving both methods to improve the safer and the more effective gene therapies here. So applications of the gene delivery systems. So far we have learned what gene delivery is and how genes are delivered. So now let us see where this technology is actually used. So point to the application image one by one here. First application is the treatment of genetic disorders. So many diseases occur because of the defective gene inherited from parents. For examples includes hemophilia, thalassemia, sickle cell anemia, cystic fibrosis. So instead of treating the symptoms throughout the life, scientists try to replace the defective gene with a healthy one. So this is called the gene therapy. Example, a child with thalassemia needs repeated blood transfusions. So imagine if doctors could deliver a healthy hemoglobin gene into the child's bone marrow cells, so the body could start producing the healthy red blood cells naturally here. So that is the goal of the gene therapy. And second application is in cancer treatments. So cancer develops because genes controlling cell growth becomes abnormal. So scientists can deliver the gene that stop cancer cell growth and kill the cancer cells and help the immune system recognize and destroy the tumors. So instead of chemotherapy affecting the whole body, so the gene delivery targets only the cancer cells here and reducing the damage to the healthy tissues. Okay. CRISPR gene editing. You may have heard about the CRISPR-Cas9, so often called the genetic scissors. So CRISPR can cut the defective DNA and replace it with the healthy DNA. So but CRISPRs also need a delivery system to reach the target cells here. So without gene delivery, the CRISPR cannot work. And the fourth application is vaccines. So one of the most and best real life example is the COVID-19 mRNA vaccines. So the vaccine contained the mRNA which was protected by the lipid nanoparticles here. So these nanoparticles deliver the mRNA into our cells, which then produced a harmless viral protein. So our immune system recognizes this protein and develops immunity. So can anyone tell me which non-viral carrier was used in the mRNA vaccine? So the answer is the lipid nanoparticles. Regenerative medicine. So the gene delivery is also used in the tissue engineering. So the scientists deliver the growth factor genes to damaged tissues to help regenerate. Example like in bones, skin, cartilages, and the heart muscles. So the gene delivery is not limited to one disease. So it has applications in the genetic disorders, cancers, vaccines, tissue repairs, and biomedical researches. So future of the gene delivery systems. So now let us look into the future. So the future of the gene delivery is extremely exciting. So AI help the scientists design the better vectors. AI predicts which vector will work best, which cells should be targeted, and which delivery method is safest. And nano robots. So imagine the robots smaller than a human cell. So these nano robots could travel through the blood vessels carrying the therapeutic genes here. They could identify the diseased cells and deliver the genes exactly where needed. And personalized medicines. So every patient's DNA is unique. So in the future, treatment may be designed specifically according to the individual genes. So this is called the personalized medicines. and smart nanoparticles. So the scientists are developing the nanoparticles that release the genes only when they reach the diseased tissues. So this reduces the side effects and increases the treatment successfully. Gene therapy will complement the conventional treatment, especially for the genetic diseases. Conclusion. So now let us summarize the today's lecture. Gene delivery is equal to the safe transport of the therapeutic genes into the target cells. So we learned the definition, need, viral vectors, non-viral vectors, chemical methods, physical methods, applications, future developments. So remember these three important points. First, so genes are like instruction manuals. If instructions become defective, diseases occur. Second, so the gene delivery systems transport the healthy genes safely into the cells. And third, choosing the right vector is extremely important here. So high efficiency is useless if safety is poor. So likewise, so maximum safety is not enough if the gene cannot enter the target cell. So the scientists try to balance the efficiency and safety also. So the gene delivery is changing the medicines from treating symptoms to treating root cause of the diseases. So instead of asking how can we control the disease, the scientists now ask how can we correct the defective gene. So this is the future of the medicine here. So before we finish, let me ask a few questions here. So what is the gene delivery? Name any two vectors. Name any two non-viral vectors. So which vector was used in the COVID-19 mRNA vaccines? So which physical methods use the electricity? So which viral vector is considered the safest? So if you know the answer, please just comment in the comment section. So I hope today's session helped you understand the gene delivery systems in a simple and a practical way. So remember one sentence throughout your pharmacy career. So the success of the gene therapy depends not only on the therapeutic genes but also on delivering the right gene to the right cell at the right time. Thank you for your patient listening.